Dear students, now we will see the diagrammatic view of translation so that the process will become more clear to you. I hope the theoretical aspect is clear. The importance of diagram cannot be ignored because when the question is supported with the help of a diagram in the paper, then it becomes very impressive, and the examiners, even if they don't want to, they'll give you marks if you'll explain your whole idea in the form of a diagram. It will give a bird's eye view of the whole procedure. Now the translation in the form of a diagram. First of all, I am going to draw the diagram of ribosome again so that you will get the concept how a ribosome looks like. So this is the small ribosomal subunit, and this is this one is large ribosomal subunits. The large ribosomal subunit has three parts, as I have told you: the A site, the B site, and the E site. Now A stands for amino acyl, P stands for peptidyl, and E stands for exit. Now the first step was initiation. Let us see what happens in initiation. First of all, there is small ribosomal subunit. Okay. The small ribosomal subunit has ribosomal RNA. The ribosomal RNA is present in small ribosomal subunit. Here will bind the messenger RNA. The messenger RNA will have its nucleotides. In the form of codons. And let us suppose the first is AUG, which is the codon. An initiation complex, a protein you can say, this is initiation factor. It will bind to at the site AUG. As soon as it binds to AUG, there will arrive a transfer RNA. The transfer RNA will have the amino acid and the anticodon AUG, so it will have UAC accordingly. So UAC will be the anticodon. This initiation factor will be here. Now in the next step, which is the elongation step, what will happen? The large ribosomal subunit will bind here. It's A site, B site, and E site. Now what will happen, I will draw again the small ribosomal subunit. As soon as this initiation complex formation is completed, the large ribosomal subunit will bind with the help of a chemical bond. The large ribosomal subunit, the A site, the P site and the E site. Now this first codon was copied, so RNA moves a little bit further and another sequence is exposed at P site. Let us suppose this is the one mentioned in the book is CUG. Let us suppose this is CUG. Now when this will be exposed to P site, a transfer RNA, the first one is still attached here. Okay. Sorry, this CUG will be exposed here. The first already existing transfer RNA is here with methionine, chemically modified methionine. When the CUG is read here, then another transfer RNA will come here. This transfer RNA will have the anticodon GAC. It will have another amino acid. Let us suppose here it is leucine. It will bring another amino acid. When it will be attached here, there will be formation of a chemical bond. Chemical bond formation, and as a result what will happen, let us draw it here. Now I am not showing messenger RNA, I am just showing the transfer RNA's reaction of amino acid. Now this amino acid will move here. Methionine will move to the second transfer RNA. This was leucine and this is methionine. So this methionine will move to the leucine. This whole step is continued. Now this empty transfer RNA, as the whole ribosome will move in this direction and the messenger RNA will move in this direction. So as this moves in this way, this will come in exit site, the empty transfer RNA. As the ribosome moves forward, the empty transfer RNA will be in empty site. So it will be in exit site, so it will be removed from here. This will arrive in the P site, and when another transfer RNA will come, it will shift its whole complex to that new transfer RNA, and then this one will also be exited. So in this way the whole polypeptide chain is synthesized. Now the termination. Now the transfer RNAs are coming and they are receiving the amino acid chain from previous transfer RNAs, and the empty transfer RNAs are exiting. This elongation will continue in the same fashion until the chain termination codon or the non-sense codon. For example, here arrives UAA. Now here we have a transfer RNA at P site with the polypeptide elongated chain, and here we have the messenger RNA. Here comes UAA. This is the stop codon. Sorry, it will be here at A site, UAA. at A site. When this will come, there is no tRNA which will recognize this factor, so release factors will come and they will release this whole polypeptide chain. The complex which is formed with tRNA, the messenger RNA, the ribosome, the complex will break and the whole polypeptide chain is released. Let me revise this for you. First of all, we studied about the structure of ribosome, the small ribosomal subunit and the large ribosomal subunit. The large has 3 sites: the amino acid site, the P site and the exit site. First of all, the messenger RNA binds to the small ribosomal subunit with the ribosomal RNA. The initiation factor recognizes the first codon, and as soon as it recognizes the transfer RNA with complementary sequence UAC will come here and it will bind at the P site. In elongation, when the next codon is exposed here, this will be read by A site. A site will call for another transfer RNA. The transfer RNA will come with amino acid leucine and it will bind here. There will be a reaction between these 2 amino acids and this amino acid will move with the leucine. The methionine will be attached to leucine and the ribosome move forward. As it moves forward, the tRNA automatically gets into the exit site and this empty tRNA is removed, is exited. The new tRNAs continue to come and the growing chain is shifted to, always it is shifted to the new transfer RNA and the old one is removed or exited. When the nonsense codon arrives, there is the termination. The release factor, it will recognize this sequence and it will break the whole complex and the polypeptide chain is released. I hope the diagram is clear to all of you. That was all about translation. In the next video, we will talk about mutations. Stay tuned, keep watching office.com.pk. Thank you so much for your time. Allah Hafiz.